Scientists put tardigrade proteins into human cells, and here's what happened. Freezing them, heating them, blast them into empty space with survival skills unlike any other organism on the planet, those hardy critters known as tardigrades will only come back for more. While it's clear their ability to withstand stress is in part due to their ability to turn their insides into gel, the mechanisms behind this act of metabolic preservation have not been made clear yet. A new study led by researchers from University of Wyoming found that expressing key tardigrade proteins in human cells slowed metabolism, providing critical insights into how these virtually indestructible invertebrates can survive under the most extreme conditions. The team focused on a particular protein called CAHSD, already known to protect against extreme drying and desiccation. Through a variety of methods, the researchers showed that CAHSD transformed into a gel-like state when under stress, keeping molecules protected and protecting against drying. This study provides insights into how tardigrades and potentially other desiccated tolerant organisms survive drying by making use of biomolecular condensation, write the researchers in their published paper. Beyond stress tolerance, our findings provide an avenue for pursuing technologies centered around the induction of biostasis in cells and even whole organisms to slow aging and enhance storage and stability. Tardigrades have already been shown that they can survive hot and cold temperatures and high levels of radiation that would be fatal to human beings, and long periods without any water, normally so essential to life. They can even survive in space. Previous research has revealed an impressive number of tricks that tardigrades use to stay alive, built up to over hundreds of millions of years. Essentially, they're very good at slowing the process of life right down to with the help of CASD, and that could be useful in human cells too. Amazingly, when we introduce these proteins into human cells, they gel and slow down metabolism just like in tardigrades, says molecular biologist Sylvia Sanchez Martinez from University of Wyoming. And when you put human cells that have these proteins into biostasis, they become more resistant to stresses, conferring some of the tardigrade's abilities into the human cells. Some way down the line, and we may be able to figure out how to pass some of the amazing tardigrade resilience into our own cells and tissues, potentially slowing biological aging and helping in treatments where safe cell storage at cold temperatures is vital, such as organ transplants. It's going to take plenty of future research to harness this transfer of abilities, research that is already underway with some studies looking at whether tardigrade proteins can stabilize important blood products used to treat genetic diseases. Early signs are promising in several areas, including the way the proteins are smartly activated when environmental stress is present and deactivated when it is not. When the stress is relieved, the tardigrade gel dissolves and the human cells return to their normal metabolism, says University of Wyoming molecular biologist Thomas Boothby. The research has been published in Protein Science, and this is on Science Alert by David Neal. Please leave your comments, and thank you for your support. Kindly support my Patreon account. The daily posts are 5 videos daily, and they are totally different from what I have on my YouTube channel. Thank you so much for your support and that you find all my content so interesting. You'll find the Patreon account details in the description box below.